The enzyme-linked immunosorbent assay, or ELISA, is an extremely sensitive test that is used to detect antibodies or specific antigens. The direct ELISA is a test for the presence of antigen. In this procedure, a known antibody is adsorbed to the inside of the well in a microtiter plate. After rinsing to remove excess antibody, the sample suspected of containing the antigen is added. Next, an enzyme-linked antibody that can react with the antigen is added. If antigen is present in the well, the enzyme-linked antibody binds to it and is retained. The colorless substrate for the enzyme is then added. Development of color indicates the presence of the antigen. In the indirect ELISA, antigen is added to the microtiter plate well, and the antigen attaches to the walls of the microtiter plate. After rinsing to remove excess antigen, the serum suspected of containing the antibodies is added. Enzyme-linked antibody capable of reacting with the constant region of other antibodies is then added, followed by addition of the colorless substrate. Development of color indicates the presence of the antibody being identified.
So in the indirect ELISA, again, we're looking for indirect evidence that the patient had been exposed to the antigen, to the infectious disease. And in this case, it's a lot of times used for HIV screening. So in well A, we put down the antibodies. In well B, we put the antibodies, or known antigen is adsorbed to the wells. So it's attached to the wells and then washed away, anything that doesn't stick. And then you add the patient serum. So the patient serum in this case does have antibodies in well A that do attach, and well B does not have antibodies that attach to the antigen. They're not specific to the antigen. So they get washed away, but the well A, they still have the antibodies present. So then when you put the secondary antibody, which binds to the FC region of the antibody, it is an antibody against an antibody, and it contains an enzyme link to it. It will attach to any antibodies that are left in the well. So there are no antibodies here for the secondary antibody with the enzyme attached to it to stick to, just antigen. And so when you do the rinse, then those antibodies wash away, and when you put this substrate in there that's colorless, it turns to a colored product, where in the well B, where there are no antibodies and no enzyme left, when you put the substrate in there, then no reaction. So the color change indicates a reaction, and the no color change means you do not have the antibodies in the blood. So a lot of times it is a yellow color that you get. We did one which was a green color in lab, but sometimes it's also red. So there are various different colors, but a color change would indicate that the patient's blood had the antibodies for the antigen. That's what an indirect test does. It tells us indirectly that the patient has been exposed to the antigen. In the direct ELISA, we call this a sandwich test because it's known antigen, known antibodies are adsorbed to the well. So you put that down first and incubated with an unknown antigen. And if the antigen-antibody complex forms, that will attract a secondary antibody which has an enzyme, and you get a color change in that well, and that indicates a positive result. So it kind of looks like this, where you have the primary antibody lined up in the well and or on a membrane, which is what we see with the lateral flow test. They oftentimes have a row of control and a row of test antibodies. When the antigens come across that, they attach, and then a secondary antibody is attracted to this antigen-antibody complex. And if they have a color tag to them, if it's not an enzyme but a lateral flow test, they'll have these antibodies all lined up, and they all have this pink or red or whatever color tag it has attached to it. And several of these antibodies all close together will cause it to show up as a line. And so you get a red line in our group A strep test. This is how it worked. So again, here you have the known antibody the unknown antigen if it is positive for this antigen. If the antigen is what we're looking for, then a secondary antibody will also bind to it. And the secondary antibody has an enzyme attached to it. So when you put the substrate in the well and the antibodies are still there with the antigen and the enzyme, then it will turn colors. And so this is what those 96 well plates look like. Usually have one row that is a positive, and I don't see a negative control, but usually one row is a positive control, so you know that the test has been done correctly and that it is ready to be read. The immunochromatographic lateral flow tests, you can see our group A strep test. It had a control line and a test line, and those were similar to the direct ELISA because it had the antibodies all lined up along the membrane. And then you stick the swab in here with the reagent. The reagent rips off the antigens from the swab and runs down the membrane. And as it pulls the antibodies with it, some of them are attached to a color pigment, which makes the red line or the blue line. So this is very similar, and if you haven't seen the Binox kits, inside there is a place, there's like this little strip of paper, and you put the reagent on the paper. It's like 6 drops or something like that, and then you swirl this little swab in here 3 times, something like that, and then you close the two things come together. The swab comes into contact with the strip which has the antigen or the reagents on it, and then it runs up the strip and pulls the antigens with it, and that's basically how it works. It looks like a little lollipop. So this is the Binax test for the COVID antigen, and it's something that they have at home. I wish these were like more like $1 a kit rather than like $10 a kit or whatever they are. But we're just not quite there yet. But I think that in the future, this will get to be a lot cheaper. So if you're concerned, you'll be able to just do a test and say, okay, well, I'm not, if I am sick or if I have the antigen, I'm not antigenic enough to create a positive line on this test, which means that it's okay for me to go and do what I need to do. So there are also in vivo tests, and in vivo tests are ones that are similar to serological. They introduce antigen or antibody to the skin and you get some sort of visible reaction. This is the tuberculin skin test is actually a result of, it's a hypersensitivity. It's a type 4 hypersensitivity, but that getting a red raised thickened lesion would indicate that you had exposure to tuberculosis. So that's another type of test, but this is more in vivo, not in vitro. So specificity versus sensitivity. One last little thing about the immunological tests before we do the concept check, but specificity means it's specific. Makes sense, right? So that's a positive or a property of the test to focus on only a certain antibody or antigen. So it's a property of antibodies and antigens that they are specific to each other. And it does not react with an unrelated or distantly related antigen. So it has to be very specific. It does not react to unrelated or distantly related antigens. And so the degree to which they define specificity, this is kind of like your definition here. So specificity is defined as the degree to which a test does not falsely detect people who do not have the condition. So no false positives. That the degree to which you have no false positives is a measure of the test called the specificity. So we also, when we look at these tests, call this a sensitivity. We have a term that defines for the test. It's a trait of the test, and it's a degree to which a test will detect every positive person. Now, so that is going to be hopefully very high, but it doesn't have to be 100% if the level is so low. So it detects, it's the measure of the detection, and that where you have even minute quantities of antigens or antibodies in a specimen. So we don't necessarily need teeny tiny minute quantities if they're not able to actually cause disease. So a lot of testing happened early on, and they rejected the tests because they weren't sensitive enough. And we don't need it to be 100% sensitive. We just need it to be sensitive enough that it detects the disease when somebody is able to transmit the disease. So this, again, reflects the degree to which a test will detect every positive person. And is that 95% of the time, or is that 90% of the time? So concept check 17.4, and I realize we didn't go through all these sections, so this is really just our second section, but agglutination tests rely on blank blank, while precipitation rely on blank molecules. So agglutination relies on whole cells, where the precipitation relies on soluble molecules. So it's not whole cells, just molecules, and they're soluble. But when they come together with the antigen and the antibody, then it's insoluble, and that's why you can see it as a precipitate. So the Western blot is used to confirm the diagnosis of HIV. That's one of the examples where we use the test. And it's like you have an ELISA test, and that's showing you that you have HIV, but then the Western blot is used to confirm the diagnosis of HIV. A pregnancy test is an example of a blank testing, and that would be what? Immunochromatographic or lateral flow tests. And blank is sandwich or indirect ELISA, detects the presence of antibodies in a patient's serum. Is that the direct or indirect? Sandwich or indirect ELISA? And so that would be the indirect ELISA. It's indirectly detecting the presence that the patient has actually been in contact with the antigen or the agent. So genotypic methods, I'm going to talk a little bit about PCR, mainly because this is one of the tests that's used for COVID, and I think you need to understand how that particular test works. So there's an animation to a YouTube link that I'll show you here in just a minute. But the PCR is polymerase chain reaction, and what it is, it's a genetic copy machine. And so it makes copy after copy after copy. Now, if you have a whole book, do you need to copy the entire book? No, you just have like maybe one page you need to copy. And so just like the DNA, we don't need to copy every strand of DNA. We don't have to copy every chromosome. Maybe we just need a bit, a little piece of one particular chromosome. And so what this does is it copies a very specific part, and it makes many, many copies of DNA or RNA within hours. And so it goes on an exponential copy method. So one copy makes two, and then those two copies become four, and four becomes eight. And then it goes at an exponential rate to where you have 30 copy cycles. You have 1 billion copies of this piece of DNA. And it can be used for a lot of different things. So it's our kind of our main test that we use in the laboratory anymore, because it's so easy and you can get these results within hours. But it can amplify them to get something, improve the sensitivity. So if you even have just a tiny bit of something, you can amplify it enough that it can be detected. And they can use it on bacteria, they can use it on viruses, they can use it on any kind of organism, eukaryotic, prokaryotic, or even a virus. And it's necessary to do metagenomic analysis of the human body. So when they were doing the microbiome tests, they were using PCR to amplify the DNA so they had enough copies of it to detect it. So RT-PCR means real-time reverse transcriptase PCR, and it uses reverse transcriptase enzyme to make a complementary DNA strand from the RNA, and then uses that DNA strand and it amplifies the DNA with a DNA polymerase called Taq polymerase. from actually a bacteria cell that lives, or bacteria that lives in very aquatic environment. It's very warm aquatic environment, and so it's adapted perfectly to go between these 94 degrees down to 72, down to 57 or whatever. There's like 3 different temperatures that it has to be present and it has to work perfectly at. And this particular polymerase does that. But that amplifies the DNA. And basically, what the real-time part means that when you get enough copies present, a fluorescent label is detected and you get a positive result. Now, how many cycles it takes for you to get that positive result is called the CT value. And the CT is the cycle time or whatever. It's just the number of cycles that required to get a positive result. And that gives you a quantitative as well as qualitative, not just that you are positive, but how positive, like how many, is it weakly positive, or do you have a lot of the antigen? Do you have a lot of the virus in you? And so if you have a CT value of less than 35, then that means possibly. If it's less than like 25, that's probably a stronger, because that means you had to go through fewer cycles before you got a positive result. And in fact, I'd say probably a CT value of 35 is probably weakly antigenic, because that means it took a million, billion copies before you actually got a positive result, something that was amplified enough that you could see it. And actually, that could indicate that you didn't have a positive result. It's a false positive. It could have just been amplifying the mRNA that was present in your body and not the RNA from the virus. And so anyway, that is basically the idea behind PCR, polymerase chain reaction. And it is a phenotypic method, or sorry, genotypic. This is a genotypic method. So that's all we're going to talk about in section 5, is just this PCR test. And so I'm going to show you this animation on YouTube, which you can link to here.